Hello everyone. Today I'm going to talk about a paper that investigated whether select species in the microbiome of bacteria influence an individual's susceptibility to depression and anxiety. In the paper they also investigated whether or not these same species inhibit neurogenesis, but I'm not going to get into that side of the paper because the video will be too long. In case you're curious, neurogenesis is the phenomenon in which new neurons grow in the brain. First of all, I'll talk about some of the methods that they used in this paper, so you've got an idea of how they did it. And after that, I'll go through each of the different behavioural assessments that they conducted on the mice and tell you the results of them. So they had two groups of mice. One group of mice received a fecal transplant from a group of healthy individuals, and within the fecal transplant would have contained these individuals' microbiome. And another group of mice received a fecal microbiome transplant from individuals with major depressive disorder or major anxiety disorder. And before they received these treatments, all of the mice were treated with antibiotics to kill the existing microbes in their guts. The next thing they wanted to find out is which species colonize the respective groups. The group of mice that received a fecal microbiome transplant from healthy individuals and the group of mice that received fecal microbiome transplants from individuals with major depressive disorder. The way they did this is through 16S rRNA sequencing, and that's just a way to identify and compare bacteria species within a sample. From now on, I'm just going to refer to the group of mice that received a fecal microbiome transplant from healthy individuals as the healthy mice, and I'm going to call the other ones the depressed mice, just to save time, because saying it's just a mouthful. But anyway, so what did they find in this 16S rRNA sequencing? They found 745 unique operational taxonomic units in total, 124 of which were unique to the depressed mice, and 178 of which were unique to the healthy mice. And all an operational taxonomic unit is a group of sequences of a particular gene, in this case 16S rRNA, that are very similar to each other. One operational taxonomic unit either encompasses a single species or multiple species, and they perform very similar functions. What they found is that depressed mice had a depletion in 14 species belonging to the Ruminococcus genus and Akkermansia genus, and they also have an enrichment of 13 species belonging to the Bacteroid genus. Now, this data is great because it, first of all, tells us that there is indeed a significant difference between the composition of the microbiome of the depressed mice or depressed people and healthy individuals. And furthermore, it describes specific differences in the relative abundance of different species between people with major depressive disorder and healthy people. So this information is quite handy because it means now we've got targets to research. We've got specific species that we have an idea could play a factor in depression, and now we can go and research their metabolic functions and find out if they do indeed directly affect depression, or if there's another factor involved. And in this case, maybe correlation doesn't prove causation. The first behavioural assessment they conducted was an open field test. So they had a 40 centimetre cube box, camera above it to track the mice's movement, and they put the mice in there and they assessed them on 3 parameters, which were namely the number of entries into the centre region of the box, the amount of time spent in the centre region of the box, and the total distance moved in the centre region of the box. And what we're assuming here is that mice who are less anxious are more likely to enter the centre region and more likely to spend more time there. And what they found was that depressed mice spent significantly less time in the centre, they entered the centre significantly less, and they travelled a significantly less total amount of distance in the centre. The next behavioural assessment they conducted was an elevated pulse maze test, where you've got a maze with 4 arms, 2 open and 2 closed. The mice were placed in the centre of this maze, and a camera again was above them, tracking their movement and timing them, and the time spent in the open arms and the time spent in the closed arms was measured, with the assumption being that mice with high levels of anxiety will spend less time in the open arms. And what they found was that the depressed mice spent significantly less time in the open arms. The next behavioural assessment they conducted was an unpredictable mild stress test. So what they did over a period of time, they randomly subjected the mice to mild stress, like a sudden change in light level, or they moved them into a different room, or a sudden loud noise, something like that. And one of the stress tests they did was a tail suspension test. So they taped the mice by the tail, so they're hanging and they can't escape. And then they left them there for 2 minutes to acclimatise to their situation. And then for 4 minutes, they measured how much they moved about. And it's assumed that mice who try to escape for longer, who move about more, are less depressed, and mice who give up quicker are more depressed. And what they found is that the depressed group of mice gave up faster than the healthy group of mice. Now, if you remember from earlier, the depressed mice had a greater abundance in 13 different species belonging to the Bacteroid genus. And as a result, the researchers also wanted to investigate 4 select species in regards to their effects on anxiety and depression. They had 5 groups of mice, 1 control which had the 4 species pasteurised as a treatment. So they were dead, they were burned alive, they were dead versions of the species. And then they had 4 other groups of mice, each treated with an individual species of bacteria. After confirming that these mice were indeed colonised by their desired bacteria, they then conducted an open field test and a tail suspension test on each of them. And what they found initially was that these select bacteroid species had no effect on their behaviour with respect to depression and anxiety. So they decided to do an ultra unpredictable mild stress test for 7 days, because this is known to produce depressive behaviour. After the unpredictable mild stress test, they conducted another open field test and another tail suspension test. And what they found initially, what they found in the open field test is that 2 of the groups of mice spent significantly less time in the centre region of the OFT. However, in the tail suspension test, the 2 same species, along with a third one, spent more time compared to the control trying to escape, indicating less depression. So we've got conflicting evidence here. So maybe they chose the wrong species, all the 13. Or maybe there's an interaction effect between the different species. Now let's talk about the conclusions and the discussions that we can take from this paper. So first of all, to speak more broadly, there's been a lot of evidence recently to suggest that the microbiome is associated with major depressive disorder, and especially this increased abundance of bacteroids that seems quite common among people with major depressive disorder. And something this study's shown is that the symptoms of major depressive disorder are transferable to mice via a fecal microbiome transplant from humans. Now, what don't we know? I think this is the most exciting bit, because it gives us a glimpse into our future. And what we don't know is the how, like the underlying mechanisms by which the different composition of bacteria in the microbiome cause the symptoms of major depressive disorder. We don't know how a lot of the species individually interact with us, the host, to perhaps influence disease in their lacking, or in their having too much of them. And furthermore, we don't know how these different species might interact and how the effect of their interaction might have on us, the host. And with that, I think I'll leave you. Thank you for watching. I appreciate your time and attention a lot. That's very kind. And yeah, like, comment, subscribe if you find it interesting. Peace out.